What is GABA? GABA is a short name of gamma amino butyric acid. It is the chief inhibitory neurotransmitter in the developmentally mature mammalian central nervous system. I have colored the word neurotransmitter here because I will explain what is actually neurotransmitter to better explanation of the effect of GABA, how does it work, and what its effect on the brain. Its main role is reducing the neuronal excitability throughout the nervous system, and the neurons which produce GABA as their output are called the GABAergic neurons. As we see here in the picture, the neurotransmitter, we know the function of the nerve is to carry the response and stimuli from one part of the body to the brain and from brain to the body. And this function is actually done by the nerves, and nerves actually transmit the signals from one neuron to another neuron, and between two neurons there is a gap, there is a junction, I mean the joint between two neurons is actually called the synapse. And the neurotransmitters are different types of chemicals which are released from the previous neurons into the gap junction, into the gap which is called synapse or synaptic cleft. And after releasing those chemicals, they attach on the receptor of the next nerve, and after attaching they exert some kind of changes in the chemical gradient that are responsible for transmitting the signals, electric signals from one neuron to another neuron. Now I will explain how the GABA is synthesized. The principal precursor molecule of the GABA is the glutamine, as we see in the picture. With the help of the enzyme glutamine synthase, glutamine is transformed into glutamate, and with the enzyme glutamate decarboxylase, it is transformed to GABA, gamma amino butyric acid. Here, one thing is to mention that the vitamin B6 and magnesium is necessary for this reaction from glutamate to GABA transfer. These vitamin B6 and magnesium presence of these two the vitamin B6 and magnesium is necessary. If these are absent, then this reaction will not be happened. So after converting to GABA, it binds with the receptor, but it's interesting to know that the synthesis of GABA is actually it's in a cyclic way. It has a cycle of synthesis. Like after this reaction, when GABA is formed in the neuron, so we can begin here the GABA here in the neuron, the previous neuron in the synapse of the previous neuron, which is called the presynaptic neuron. And after releasing, when the nerve signals needs to transmit the signals, GABA is released. After releasing in the synapse, they attach on the receptor, and after exerting its function, what is happening to the GABA? What happened? It is taken up by some kind of cells in the brain which are called the glial cells. After taking up into the cell, the cell's mitochondria secrete an enzyme which is called the GABA transaminase. With this enzyme, the GABA is transformed into the glutamate, and with the enzyme glutamine synthetase, as we see in this picture, that this glutamine and glutamate, this reaction is reversible. Glutamine can be transformed into glutamate, and glutamate can be transformed into glutamine, and that's happened in this glial cell. And after with the enzyme, then glutamate is transformed into glutamine. And glutamine, there are some kinds of transporter in the neuron and also in the glial cells, which are called the glutamine transporter. With this transporter, glutamine is transported into the neuron. So it is transported into the neuron. And again, after transporting the glutamine into the neuron, again this reaction is continued. So glutamine is transformed into glutamate, and glutamate is transformed into GABA. That's how. That's why it is called a cyclic way of GABA synthesis. After synthesizing, it is not lost. It is taken up by the glial cells, and it is again, with the help of the enzyme, it is transformed into glutamine, and glutamine is again transported into the neuron, and it is transformed into GABA, and this cycle continues to exert the inhibitory effect of the GABA. How the GABA act or exert its function, its inhibitory function, as I have mentioned in the previous slides, that the GABA main function is inhibitory neurotransmission. I mean, it's inhibit the neurotransmission, inhibitory neurotransmitter. So it is a neurotransmitter that inhibits signals transmission. So after releasing in the nerve cell, in the synapse, as we see here, the GABA is released in the synapse. It binds to the plasma membrane receptor of the pre and post, especially in the post-synaptic neurons. After binding with the receptor, it helps to open the ion channels. Ion channels, you have to know that there are different kinds of ion channels: ligand-gated ion channels, voltage-gated ion channels. I think we can discuss about those ion channels later, but right now just know that the GABA binds to the receptor and helps to open the ion channels. In the ion channels, some channels helps to transmit, pass the different kinds of ion, but GABA helps to open the chloride ion channels. So chloride ion goes inside of the cell, and the potassium ion goes outside of the cell. Influx of the chloride ion and efflux of the potassium ion. So chloride ion going into the cell and potassium ion going out of the cell. In this way, it causes the hyperpolarization of the cell. So hyperpolarization, the word is somewhat quite new, maybe quite new, or you may not know what is hyperpolarization. Actually, this word is related to a most interesting but somewhat quite difficult topic, which is called the action potential, resting membrane potential. Now we will try to discuss about something about the membrane potential. First, I want to make a definition of the hyperpolarization. Hyperpolarization is actually a change in a cell's membrane potential that make it more negative. So, change in membrane potential. What is the membrane potential? We have to know. In the resting condition of the cell, we know the cells are continuously working, but cells also have some resting period. Between two working period, there are some resting period, which is at that time, during that time, cells outside and inside of the cells, there are different chemical concentration, ion concentration. Like if we see in that picture that there are different types of ions: sodium, potassium, calcium, magnesium, chloride, and so on. And we see that there are different concentration gradient inside and outside of the cell. And sodium ion outside of the cell is much more higher than the inside of the cell. The potassium ion also higher in the inside, in the cytoplasm, and very much lower in the extracellular fluid, outside of the cell. Same as the chloride ion, which is the most relevant in our present topic. The chloride ion, we see here, it is very much higher in the extracellular fluid than the intracellular fluid. So what happened during the resting condition? In the resting condition, the concentration gradient between inside and outside of the cell makes the plasma membrane negative. It has a negative charge. Polarized. Polarized means, we know the polarized. Polarized means there will be positive and negative in two pole. So in outside of the, in case of cell, this concentration gradient make inside of the cell is negative and outside of the cell more positive. So if we insert a probe for measuring this electrical, measure the electric signals, it is minus 70 millivolt. This is the condition in the resting membrane potential. But when cells needs to stimulate, the nerve needs to signal, transmit the signals, it needs to be stimulated. So for stimulation, it needs to be depolarization. The polarization should not be present there. It has to be positive. So in that case, when nerves needs to transmit the signals, it becomes depolarized, from polarization to depolarization. So in that case, some channels become opened, which are called the sodium and potassium channels. In that case, sodium goes inside of the cell and potassium goes outside of the cell. But 3 sodium goes inside of the cell, 2 potassium goes outside of the cell. In that way, the negative charge of inside of the cell become positive. And ultimately, both inside and outside of the cell, there will be neutral charge, which is in the depolarization we see here the 0 millivolt. But what happened in the GABA? So if the chloride ion goes inside of the cell, so we see here this is the resting phase, this is the resting condition. Inside is negative, outside is positive. If chloride ion goes inside of the cell and potassium goes outside of the cell, which is actually happened in the GABA after attaching to the neurotransmitter receptor of the next neuron, the chloride goes inside of the cell. That means chloride is carrying more negative charges inside of the cell. So it will be more negative. In case of polarization, it will be more negative, means more hyperpolarized. That's why when the GABA acts on the neuron, it is called hyperpolarization. Hope you understand why it is called hyperpolarization. So we see here in the picture, it will be more clear that during the resting phase, it is minus 70 millivolt. When cells needs to be stimulated, it tends to, the inside of the cell tends to be more positive. So the difference between outside and inside of the cell will be more laser. That will be at a point of time, it will be neutralized, and even maybe it will be positive if the cells needs to be more stimulated. But in case of hyperpolarization, this curve goes down of the minus 70 millivolt. That means this is called the hyperpolarization. This level is polarization. When it goes down, it will be hyperpolarization. If it goes up, it is depolarization. And that's how the GABA works. Because GABA carrying the more negative charges inside the cell, that makes it hyperpolarized. And if it is hyperpolarized, it inhibits the nerve signal transmission. That's how GABA works. Hope you understand and liked my video. If you like it, please subscribe.